Okay, so hearing is when the auditory system detects sound waves that can change and vary in intensity and frequency. So it goes from the outer part of the ear, which stimulates the inner part of the ear. And this is when mechanical energy is transduced into neural activity, where action potentials can fire in the brain to send signals to the rest of the body. And the ears also filter out unnecessary noises. So the external part of the ear, the part of the ear that people can see, is called the pinnae, which funnels sound waves into the second part of the ear, known as the ear canal. So the pinnae can enhance or suppress sound waves through ridges and valleys in its structure. The shape of the human ear increases reception of sounds between 2,000 and 5,000 hertz, and hertz is the sound wave measurement. So the middle ear links the auditory canal to the neural receptors and is made of tiny bones. That is part of the tympanic membrane, which seals the ends of the auditory canal, and the ossicles, which is made up of the malleus, incus, and stapes, and they're really tiny bones. Sound waves cause the tympanic membrane to vibrate at the same frequency as the sound that the ear is hearing, and the vibration moves the ossicles into the small oval window. And there are two muscles that vary the mechanical linkage between the ossicles to improve auditory perception and protect the inner ear from too high-pitched or loud sounds that could damage it. So there's tensor tympani and the stapedis. So the middle ear muscles help make self-made sounds quieter, like swallowing and yawning and things like that. So the cochlea is a part of the inner ear that contains the primary receptor cells for hearing. And it has three canals called the scala vestibuli, the scala media, and the scala tympani. The width decreases along the length of the canal, and the membranes that divide them decrease along the length of the spiral. And these are filled with non-compressible fluid, so that movement inside the cochlea in response to push the oval window. The helps with this is the round window that separates the scala tympani from the middle ear. So the principal component that converts sound into neural activity is the organ of corti, which is made up of hair cells, framework of supporting cells, and termination of auditory fibers. And the base of it is called the basilar membrane, and it has inner hair cells and the outer hair cells, and they each have stiffer hair cells, which are called stereocilia. And the tectoral membrane lies on top of the organ of corti, and the stereocilia protrude into the base of it. Auditory nerve fibers contact hair cells at the base. So there's four kinds of synapses and nerve fibers in the organ of corti. Two are afferent and two are efferent. So the main neurotransmitters involved with those are acetylcholine, GABA, glutamate, and dopamine. Outer hair cells don't detect sound, but they push on the tectoral membrane in response to the brain via efferent nerve fibers, and they change length with hyperpolarization and depolarization to fine-tune the organ of corti. And inner hair cells receive messages from the efferent neurons to inhibit from afferent responses to loud sounds. Fine hair cells called top links run along the top of the stereocilia, and sounds that cause the hair cells to sway away put tension on the top links, which causes the ion channels to open. And as the hair cells sway, they quickly open and close. And opening the ion channels allows for potassium and calcium ions to rush in, depolarizing the cell. And the depolarization leads to an influx of calcium ions at the base of the inner hair cell, which leads to the synaptic vesicle fusing and releasing its neurotransmitter, which stimulates afferent nerve fibers to trigger action potentials. Hyperpolarization causes the outer hair cells to lengthen by protein prestin, and depolarization causes them to shorten. And autoacoustic emissions are sounds produced by the cochlea itself in response to environmental noise. So the vestibulocochlear nerve runs from the cochlea to the brainstem to the auditory nerve, and a tuning curve is a graph that shows how a single auditory nerve can respond to varying intensities and frequencies of sounds. And each nerve fiber divides into two main branches as it enters the brainstem, and each branch then goes to one of two groups of neurons, either the cochlear nucleus or the ventral cochlear nuclei. Bilateral input is the first stage in the CNS at which binaural effects are processed, and it's important for localizing sounds because it's coming from both ears. So the inferior cochlea is primary auditory centers in the brain, and it sends outputs to the medial geniculate nuclei of the thalamus. At the medial nucleus and auditory cortex neurons are excited and inhibited by neighboring frequencies, and this sharpens frequency responses. And all auditory pathways have synaptic organization where they are arranged in an orderly map according to the auditory frequencies that they respond to.